Hello class, this is Mr. Lehman. I want to show you the Wild Bean Lab, so in case you're a virtual student and not able to do this in class, you have some data so you can complete the activity. Alright, so inside of here, inside this bag, we got some white pinto beans. And those represent, each bean represents an organism. And we're trying to do the mark and recapture method to estimate the population size of the beans without actually counting the beans. So we'll see how accurate we are. And the mark and recapture definitely has some limitations in certain types of environments, certain types of regions, because it's just really hard to count populations, especially if you're looking at a wide, vast area. So this is one method that scientists use, and here we go. So what we're going to do is reach into the bag, and we're going to pull out some beans, a small handful of beans. And here we go, I grabbed some organisms. So these are the organisms that I captured. Here we go. All right, so I didn't capture all the beans in the bag. That's the whole population. This is just a sampling. So what we're going to do here is we're going to figure out how many beans we captured. I'm going to try to figure out how to support my iPad the best so we can count these. There we go. So I'm going to put these in groups of 10 so you can count them at home. Look at that. 3. Let me figure out my iPad where I can put these so you can see it. So each one of these is a group of 10, exactly 10. So we got 10, 10, 10. So by reaching into the bag, I collected a population, 30 individuals from the population. All right, so if we're looking at our data table, main numbers here we're looking at. Total number in the first capture, hopefully this isn't backwards for you, but we would write in 30. Total number of beans in our first capture, we captured 30. Now in the instructions, if this is real life, we would mark those animals, mark those organisms, and put them back out into the wild. But I don't want to put an X in every one of these beans, right? We're trying to limit touching different things due to COVID. So here's the idea. We're going to get some red beans. All right, we got some kidney beans, and I'm going to count out 30 kidney beans, and I'm going to replace those 30 beans. So take the 30 white beans, pinto beans, I'm going to take those out, put those aside for now, and I'm going to put 30 kidney beans back into the bag. So I'm going to go ahead and count out 30 kidney beans. These represent the organisms that were captured and were marked and put back out into the wild. So there's 20, 23, 26, 29, one more 30. So here it is. So you can see it, 30 pinto beans. Or I'm sorry, 30 kidney beans replacing the 30 pinto beans. So I'm going to... Sorry, I dropped an organism on the floor. Sorry about that, buddy. All right, so now I got 30 red beans, kidney beans in here. I want to shake it up, mix all those white beans, pinto beans with the red kidney beans. We have a whole bean medley right here. All right, so now I don't want to peek. I'm going to reach back in. This is the recapture. I'm going to reach back into the bag and pull out another small handful of beans. Right, we don't want 1,000 beans in our hand. We don't want one bean in our hand. We want a nice random sampling. And so look at what I pulled out. I pulled out some pinto beans, white beans, normal species that were not captured before. And who wants to eat this kidney bean here? That's pretty nasty looking. All right, so yeah. So I'm going to count my beans again. I'm going to figure out how many of these organisms were marked before. And sorry, it's hard for me to hold this and move it around. I don't have an iPad stand. I need to get one and borrow one. All right, so we got 8 individuals that were marked before, that were captured before, and now we got 3, 6, 9, 10, 13, 16, 19, 20, and 4 left there, 24. All right, so 8 and 24. So we'll go back to our data table, and hopefully this isn't backwards. Number in the recapture, so we had a total of 30 beans in the first handful that we pulled out. There were 32 beans in the sample, the organisms that we sampled. And then the number of beans that were marked and recaptured were 8, right? We had 8, that's the number of red kidney beans in that second handful. All right, so we had 30 individuals captured the first time around, put them back out into the wild after we marked them, captured a second sample of organisms. There were 32 organisms in that sample. Of those 32, 8 were marked, 8 were kidney beans, 8 were organisms that we caught before. So you're going to use the mathematical equations to try to figure out how big the bean population is inside this bag. And we'll see how close we are. Now, I counted this before, and there were 160 beans in here. So I'm not going to dump these all out and try to have you count them from home, but there were 160 organisms, beans, in the original population. So you need to know that. So see how close our data is, and I'm going to put together a second trial.